The 1000 Genomes Project. The 1000 Genomes Project was an effort to sequence genomes of at least 1000 individuals. The purpose of this project was to create most detailed and medically useful catalog about the human genetic variants. The 1000 Genomes Project therefore aimed to produce an extremely detailed catalog of human DNA variations that can be used in future studies of people with particular diseases. The objective of the 1000 Genomes Project was to produce a catalog of variants present at 1% or greater frequency in the human population, and ultimately to make it down up to 0.5% or lower within the genes. So as a general, 1%, and within the genes the objective was 0.5%. And to increase the sensitivity of disease discovery by 5-folds across the genome and 10-folds within the genes, provide better understanding of very rare genetic diseases. So the objective of the 1000 genomes was to provide the better understanding of the rare genetic diseases as well as common genetic diseases. The objective of 1000 Genomes Project was to understand the contribution of common variants to common diseases like diabetes and heart diseases. So what is the effect of that? What is the percentage of common variants and how common variants they are responsible for causing the common diseases? Another objective was to identify single nucleotide polymorphism, but in addition to single nucleotide polymorphism, the differences like the chromosomal rearrangements, chromosomal deletions, and duplications was also one of the objectives of 1000 genomes. Here is the history of 1000 Genome Project. Initially, Human Genome Project was sequenced by 2002. Then there was a project International HapMap that was completed till 2008. And from 2008 onward, the project launched, which is called as 1000 Genomes Project. The comparison between allele frequency of the variants and the clinical impact, that if the allele frequency of the variants is less than 1%, then there should be sequencing and it has more impact. While on the other hand, if the allele frequency is greater than 5% or greater than 5%, then there should be genome-wide genotyping. So this is the comparison between the allele frequency of the variants and the clinical impact of whether that the frequency or the variants should be identified either by sequencing or by performing the genome-wide genotyping. These are the populations which have been used in this project. The name of the project is 1000 Genome Project. So these are all populations whose DNA has been used in the 1000 Genome Project, including the samples from Pakistan. That samples from Pakistan has also been included in the 1000 Genomes Project. So if we conclude that what is the 1000 Genome Project, the objective of the 1000 Genome Project was to create the most detailed and medically useful picture of human genome variations. Variations may explain individual differences in susceptibility to diseases and responses to the drugs that are usually used by the human beings.